Hi, in this video, I present my reflection on the news story Jumping Genes Repeatedly Form New Genes Over Evolution. This article discusses a major study examining the impact of transposons on evolution, specifically how transposons create new expressed genes or new transcription factors. It was discovered that transposons jump onto existing genes or gene exons by copying themselves and inserting themselves into the gap, or by cutting themselves out of the old region and inserting themselves into a new one. This article has much in common with the content of the GEN2052 lectures, namely transposons, their types and mechanisms, the methodology of working with blasts to compare genomes, and the use of a program to find discrepancies. The study used these methods to predict or classify genes, for example. I initially considered junk DNA to be an unnecessary part of the genome and did not consider classical regulatory mechanism, but the lectures and then the articles with a real life example emphasize and complement each other. For example, there is a gene neofunctionalization, that is a mechanism whereby a new mutation arises from old information. There is also a link to gene regulation with transposons fuse with an existing gene to become transposases, meaning a protein based on the gene regulate the expression of the old genes. So here is the strengths. The article and the original study have many strengths. First of all, 500 tetraploid genomes were sequenced and analyzed. This revealed that over 100 genes are the result of fusion of all genes with transposons. In addition to the aforementioned sequencing and blast techniques, additional laboratory experiments were conducted. Specifically, one of the bad transposons gene, we call it crumbiner, identified and named in the study. And it was excised using CRISPR-Cas9 technique. Its removal resulted in the inactivation of over 100 other genes, demonstrating the potential of such fused genes as transcription factors in a live experiment. The limitation of the study, first of all, we have results from one gene study in a bat. The other findings, and I recall there are over hundreds of them, have not been verified. We only have bioinformatics data based on sequenced genome, which cannot 100% confirm the validity. Furthermore, the article in the study do not indicate in any way the influence of this gene on phenotype, which again casts up on the strong evolutionary influence of transposons. Summarizing all the pros and cons of this article, we can confirm that evolution may be a much more complex process than simply a set of spontaneous mutations. It is also the first step towards eliminating the concept of junk DNA, meaning that anything that is not currently a source of suppression still has potential. In the future, this study may inspire research into transposons and the impact on cancer and other disease associated with mutation.